science, nous on fait de la science. Nous on travaille pour la science. Pour la science.
So, hey Paul.
Bonsoir, Max.
So, time for a new episode of Tabasas, and that will be the second episode. So, let's bring our subject of today, which is what is a cell? So, for those who don't really know us personally, Paul and I are in vitro toxicologists, which means that we mainly play with cells. So then we thought that maybe it was good to have an episode on really talking about what is it, these cells exactly.
Yeah, cells are basically, this all life is a cell. So a cell is the smallest functioning unit. I think in the last episode I said it's the ability of a single unit of life to deal with the external world on its own terms. And then, well, we might get back to viruses later. That's a
the difference
different
between
form.
virus and cells, or are virus cells or not?
But to start it off, though, Max. So basically, well, just to put it in context, we also said last week that as a human being, one of the animal species on the planet, we're made up of, we don't know exactly for sure, but about 200 different distinct types of cells, but billions of them. And we have billions and billions of other cells, bacteria mostly, that live in symbiosis with us in our gut and on our skin and in our mouth and everywhere. And they mostly are symbiotic. And you found the original definition of a cell, right?
Yeah, not the definition, but how it comes.
Where the word came from, yeah.
Yeah. So I hope it's correct, because that's actually what I'm teaching my students in their very first course with me during their bachelor degree. And then I introduce them to cells and how it was discovered. So the discovery of the cells is attributed to the first person who were able to see them, actually. Because before the invention of microscope, you couldn't really see the cells. So among those people, one of them was, what's his name? Hooke? Or how do you pronounce that?
Hooke, I guess.
Yeah, maybe it's better if I spell it, because with my very nice accent, it doesn't really necessarily help. But Hooke, Hooke. And Robert Hooke in 1667 was playing with probably two pieces of glass. And by aligning them correctly, he got one of the very first microscope. And with this microscope, he started looking at stuff. One of these stuff in particular was the outside layer of, cork, right? The tree, which is made, which is used to make the top of the wine bottle. So if you slide cork very thin, then you can look at it using a microscope. And then he published an article at that time using this very basic microscope where he described what he saw. And when even nowadays, if you look at this under a microscope, it clearly looks like honeycomb. And in this honeycomb, the very tiny hexagonal shapes that you see were called and are still called cell. Cell is a Latin term which means basically a small room. So when he described what he saw in the microscope, he said, what I see is something looking like the small room. So basically the cell, which you find in honeycomb. And that's from this specific analogy that the term cells come. And from that time, 1667, every single cell, which is considered building blocks of living organisms. And these living organisms can be only one cells are called like that, cell.
Yeah, and well, we know now there was a revision of how plants, animals, and prokaryotes have evolved. So I think we dropped on this the last time as well. So the planet, our Earth, is about 5 billion years old, and we know that life has existed for about 3.5 billion years. It started off quite simple. There were single. Well, it was one type of what's called a prokaryote. So they were like bacteria, and then they diverged into another two species, bacteria and archaea. And then strange things started happening to the archaea. They started adopting things into them. The first jump, we think, was when mitochondria moved into the archaea. And I guess we can explain a little bit about mitochondria a little later. But they moved in to the archaea about 2 billion years ago. And this allowed the development of plants, animals, and fungi. And that was the origin of these three branches of life that I guess we associate quite closely with ourselves, because we have them in our house, we eat them, and we hunt them, and we have dogs and cats, and we are ourselves an animal. And then much later, actually, the chloroplast moved in. That's into the same type of organism that made plants. So cells moved into other cells to facilitate different types of life, and then at some stage multicellular life became apparent, and then became quite sophisticated. Although there is still very complicated types of unicellular life. They can be enormously complicated too.
I'm quite familiar with this endosymbiotic phenomenon, which is kind of, which explain a lot, including why those two specific organoids that you mentioned are almost the only ones with two membranes. An inner membrane, the thylakoid, or the inner membrane of the mitochondria. So thylakoid in the chloroplast, and the inner membranes of the mitochondria, and an outer membrane. And that would fit perfectly nicely with the idea that it has been engulfed into another organisms. But these two, chloroplast and mitochondria, are energy producers for the current eukaryotes.
Yeah.
What was producing energy in these eukaryotes before they acquired those?
Well, the idea was that oxygen originally was very rare in the environment. And then as cyanobacteria started, because they have chloroplasts as well, or they have chlorophyll, and they produce a lot of, they started to produce a lot of oxygen, and then this oxygen became toxic to other forms of life. And mitochondria, or the ancestor of mitochondria, used oxygen. We don't exactly know where it got the oxygen from, but there might have been a little bit of oxygen in the atmosphere. Or it might have got it from molecular oxygen being produced by other processes. And then,
isn't the oxygen on the planet the result of the activity of chloroplast?
I think, but probably 99.9%. Yeah. But I don't know. There might have been. There's lots of people who research this. There might have been really trace amounts that was enough for the mitochondria just to survive. And then when oxygen dominated, it might have been too much for the mitochondria, and they better hid in archaea. But the archaea then really could manage this oxygen better because they were using it, and it actually lowered the molecular oxygen in the archaea. Is one hypothesis. But before we go too crazy on our audience, so mitochondria are oxygen consuming they're organelles now. So they're in every single cell in plants, animals, and fungi, but in our body, so in the human body, it's in every single cell except one, which is the red blood cells.
Yeah, only in eukaryote, not in prokaryote. Not in prokaryote.
No, I didn't say prokaryotes. I think I said plants.
No,
Yeah,
You said in every single cell.
in every single cell of the plants of eukaryotes.
Okay.
Sorry, yeah. Sorry,
Of eukaryote. Yeah.
And they basically use oxygen to make the energy, the majority of the energy for the cell, and this is a form of energy called ATP. It's a really sophisticated system. They're like the mitochondria work in the inner membrane of the mitochondria. They have these proteins that transport electrons to each other, and the last protein is called complex 5, and it's basically a molecular motor. So it actually rotates and pumps protons down against or with a gradient to generate ATP from ADP. And in effect, that's how our fat is burnt, and it's actually consumed in a way, and that's how we get our energy. You get energy also from quick sources like sugar, but it's mostly from oxygen consumption. And this oxygen consumption, it produces water and carbon dioxide, and the carbon dioxide is what we breathe out. So in the atmosphere, there's, I mean, I know everybody's concerned about CO2 rising in seas and stuff like that, but compared to what's inside our bodies, which is about 5%, which we breathe out, it's very low in the atmosphere. So it's about 0.03, depending on how you measure it. So your lungs can get rid of this really quickly, and they can decide how much CO2 stays in the body. And another funny thing about mitochondria, you get it, well, we're pretty sure you get nearly all of it, or if not all of it, from the maternal line, so from your mother. Because the egg, the oocyte, the egg cell, has loads of mitochondria in, and it's like thousands of times bigger than a sperm, and so it's all there. And there's even ideas that may be the reason why there's two sexes, because mitochondria have their own DNA as well. There's kind of a dual evolution going on, that the mitochondria are looking for a good genetics. It's one theory, because if it only comes from the maternal line, in a way, the mitochondria are looking for a good mate. That they can have. They're looking for a good male to donate their mitochondria to. Is one theory I heard Nick Lane discuss that.
At one stage, I was looking at the different theory about, because obviously because a male is XY and a female is XX, so only the sperm can give an X or a Y, which means that basically by choosing whether you give to the egg an X or a Y, a male would then be responsible for the sex of the future baby to be born.
Yeah.
Right? And apparently, I guess in some philosophy, people were not that happy of having to bear the responsibility of giving birth to the sex they were not happy with. So some then research luckily came through, demonstrating that basically it was the egg which was, by modifying its shape, was selecting which sperm was making it through or not, and was somehow able to select which of the spermatozoid can make it through. So basically, it felt to me like this kind of discovery from science was just finding a new backup to say, if the baby is from the wrong sex, from what I was expecting, it's my wife's fault, not mine. So back to middle age, this girl is not good because she can only give birth to female and not male.
Although that's not true at all, right?
Yeah, I think there is some scientific things into
that.
But it's just that the science in that respect, I think, is just over-abused by people who wants to defend any idea or decide who is to be responsible for what sex is a baby, which basically I don't think matters at all.
in modern day, it doesn't matter.
No.
And isn't it pretty much 50-50 anyway?
Not really, as far as I know. Because there is also this thing that some spermatozoids, the one bearing one of the two, I don't remember whether it's the X or the Y, and I'm talking here just about the human. One of the two is having a longer half-life, which basically means that if you have sex with a girl a few days before she ovulates, then the prognostic of having one specific sex of child is getting higher, because then the surviving spermatozoid would be of majority one chromosome and not the other one.
Isn't it some reptiles or I think it's reptiles. It's temperature dependent which sex is generated of the eggs.
Yeah, that as well.
Yeah.
But
actually thinking about that, if you're just thinking, well, now we're talking about eggs and fertilization. Like one of the biggest cells you see every day is an egg. It's a chicken's egg, because I mean most people eat chicken's eggs.
Yeah.
And that's one cell. It's huge.
Yeah. How do you call this giant non-flying bird?
Ostrich.
And how do you call the people living in Austria?
Austrians.
Okay.
Yeah. They're not the same. No, there's no ostriches in Austria.
No,
or kangaroos for that matter.
No, it's just that autruche is a French name for those birds, and yeah, ostrich would sounding like good to me, but I was afraid that if I say ostrich and it's the wrong one, it would just create some more confusion into the podcast, that I would start referring to a bird and indeed kind of mention something coming from Austria.
Yeah, well, they call them. Yeah, well, Österreich is the name, and that's why it's probably in France like that. Yeah. So because in German it's Österreich. Yeah. Yeah. So
this is only one egg as well, right?
What?
This giant ostrich, their very big egg is only one cell.
Yeah.
And so probably one of the biggest one you can ever find.
Yeah, it's only the biggest thing I can think of. That's one cell.
Yeah.
I mean, there is unicellular organisms that are also really quite big. They're millimeters, but nothing as big as an ostrich egg that I can think of. Yeah. But they're special. So egg cells are always very specialized. But just thinking about all the different cell types in your body, right? So let's give a few examples. So I guess we're all aware we have a heart, and the heart beats, and that's done by cardiac muscle cells, which
that's not the easiest one to start with.
No, but it's not. No, well, none of them are easy. Tell me an easy cell.
It's just that. Well, when you introduce people to cell, you start with a kind of a general shape of it, which is it's made out of a membrane, a nucleus in the middle. It's somehow round, more or less, like an egg. Yeah. And the inside, the big stuff in the middle, the biggest thing inside it is the nucleus, where the DNA is localized mainly. And then you have floating organoids, and then you have mitochondria.
There's probably no such thing as floating organoids. The cytoplasm is not really a liquid soup. No. But okay, let's talk about the main components of all cells. Well, there's a few exceptions, but so Max, you got the nucleus, which the DNA is inside, and in eukaryotes, it's really complexly wind. I think people are aware of the double helix structure, but it's like a thread that's wrapped around histones and extremely compacted, except when the cell is doing what's called transcription, when the gene is being read. The rest of the time, it's really compacted. And then what happens is a gene, when it's being read, there's a protein that docks a transcription factor, and then mRNA is produced. And that mRNA leaves the nucleus and goes into the endoplasmic reticulum. And then there's specialized RNA that add amino acids to this. And then you make what's called a protein. And that protein can stay where it is or move somewhere else in the cell and then have a function. So the genes encode in the nucleus so they're basically the fingerprint, which are evolutionary conserved, and then the genes are read, and then they make proteins, and the proteins are, depending on what they're made of, are distributed and have function, and it's the proteins that have the function. And then the proteins basically can manipulate their local environment and change the cellular soup, which is made out of small molecules. We call them metabolites. And so we have this thing called the endoplasmic reticulum that goes around the nucleus where the proteins are made. And then in the cytoplasm outside of that, we have organelles like mitochondria and lysosomes. And what keeps the whole cell together is a lipid bilayer, which basically prevents stuff unscheduled getting out of the cell or unscheduled getting in. And then what allows things to get in and out are protein transporters within the membrane, which is basically the whole system.
Sounds good. Yeah, and I must say, it took me some time to realize how dense was the cytoskeleton, because you are first taught when you take lessons about biology and cells, this assembly of protein making the skeleton within the cells is not something you put too much details onto. And it really took me some time to really readapt my thinking to the fact that there is so much stuff inside the cells which give it its shape and that is organized within it. Because you see so many drawings of the cells where you basically see only the organoids and you don't see the inner cytoskeleton, that you tend to forget that it's there, or it takes you some time or some experience to do some staining of these proteins within it before you realize that it's fully packed.
And all the cells know basically their orientation. So this is a thing called polarity. And then depending on what type of cell they are, that polarity can do different things. So if you think of a neuron, it fires in one direction. So these are the neurons that are the majority of the cells in the brain.
Why would you say every single cell knows their polarity?
Yeah,
I think some cells knows. I don't know whether every single one of them knows.
Give me an example of one that doesn't.
Or one lymphocyte. Those lymphocytes knows.
Yeah, because they move to the site of action, so they can go in and out of cells. So they need to know. They need to be polarized to be able to move.
Why is that? No, they don't need to be polarized to be able to move.
To move in and out of tissues, yeah, they do.
Yeah, they don't need to be polarized to do so.
Well, how can they move in between tissues if they're not?
Because lymphocytes do have adhesion molecule on their periphery, everywhere around the periphery, not on a specific site around it. And then they go through the blood flow. And then in case of an infection, there is some expression of complementary adhesion molecule on the vessel. And then you have a series of interactions. The first interaction is basically slowing down the lymphocyte. Then it starts rolling on the vessel made of endothelial cells. Then it starts rolling till one of these adhesion molecules form a firm adhesion. And once this firm adhesion is made, then it starts really going across in what is called diapedes, which is when a lymphocyte leaves the vessels to penetrate the tissue. So, which means that originally these adhesion molecules are orientated all over the surrounding of the cells, so that independently on which part of the lymphocyte touch the adhesion molecules, then it will have a chance of integrating with the complementary adhesion molecule on the vessel.
Yeah,
So
but that's a
it's
form of
not originally
polarity.
polarized.
Yeah, but no, it's not originally polarized, but it can polarize on cue. You ever seen a neutrophil hunting a bacteria?
Yeah.
It looks like it's a free living organism, because it can basically molecularly identify where the bacteria is, probably to do with LPS or something the bacteria releases, and then it knows it actually moves towards it. And that actually movement is a type of polarization. So yeah, you're right. Not those cells are not always polarized, but they can immediately polarize when needed.
Because we tend to teach some histology, and when we do so, we define some cells as being polarized, basically as opposed to the ones which are not polarized when you look at them. The one that when you look at them, they are just round with a nucleus in the center. They are supposedly non-polarized, whereas the one which in particular are forming tissue, where you have an
But
apical...
do you not think the way we teach biology is a little bit outdated? I think.
Yes, to some degree, yes, but this...
Because Max, me and you know, so Max and I grow, and Max grows mostly endothelial cells. I grow mostly epithelial cells, and you can grow these cells in a cell culture dish. And if you grow polarized epithelial cells, they start off so you can make them single cells with a process called trypsinization. So you strip away the ECM and you disconnect them from each other by removing calcium. And then they're in suspension and they don't look polarized, right? But as soon as they hit the plastic, or if you've coated it with an extracellular matrix, which is the proteins that are on the outside of cells, they immediately polarize. And then because they need to be polarized because they only can work like that in a tissue.
Yeah.
And when the tight junctions form, they think it's the most obvious part of the polarization process. And then they know what's up and what's down. And all the cells, so we often grow cells in tissue densities of something like 10 million per, I don't know, what is it? It doesn't matter. In a 10 centimeter dish, you've about 10 million cells. And every single cell in that dish is facing the same way. And they work together in unison. And then if you do a trypsinization process, they'll all be depolarized again. And you can do it over and over and over again, and they will always repolarize in the right direction.
No, I tend to agree with that because I would have to double check that, but I think most textbooks define polarization due to the fact that there is tight junctions. The assumption there is that the protein you referred to previously, which are floating in the plasmatic membranes, they can basically float all around it, in and out in the inner sheet, external sheet, and they can move all around the cells, except when there is tight junctions. And these tight junctions are kind of a very important connection between two adjacent cells. And once you have that, then you define a up and a down, or at least you have something which is in the middle, assembled, which prevent basically protein from going from one side to the other side.
Yeah.
That's what the textbook. Yeah, that's what the textbook said. But I agree with you in the same time that even the single cells, as soon as it attached to a matrix, then start really changing shape.
Or chemotaxis, Max.
Yeah,
so you were talking about white blood cells. I mean, they're very chemotactic. So they know now where they want to go, and if they know where they want to go outside of the blood flow, then they can go against it. I still find, well, I mean, I think in general science is fascinating. So we say we've about 200 cell types and distinct different cell types in the human body. Let's just name a few so you get some ideas. So we said neurons, but I think that the biggest ones are the sensory neurons, right? Because they're the whole length of your body.
Yeah.
As one single cell, the whole length of your body. Then you've got, if you're female, you have the oocytes, which are pretty big. In your heart, you have several different cell types. It's mostly cardiac muscular cells, but you have these pacemaker cells as well, which basically give the electrical conductivity to know when the heart should beat in unison. I like the statement that everybody thinks we have one heart, but we actually have two. We have one that services the main body, and then we have one that services the lung. and it's just put in one unit, but they work fairly independently. So that's the heart. Then we have the liver, which is made up of 3 or 4 different types of cells. So one of the cells, the hepatocytes, are basically, they produce nearly all of the proteins in the blood, things like albumin and iron binding proteins. And they also produce what most lay people know, LDL and VDL. And these are little myocells with receptors in that just basically send fats around the body and go back to the liver and tell them how that's going. And the hepatocytes, mostly what they're doing is burning fat and breaking down fat so other cells can get it. But they're also very involved in drug metabolism, but that's almost an accidental thing. And then in your intestine, you've got loads of different sections after the stomach, which has that the bacteria is in there, and that really helps break down the food. And actually, most of that stuff goes directly to the liver after it's taken up. Then you have the kidney, which has pretty complex architecture. So you've got that's what I specialize in. So you have in each kidney, you have about 2 million nephrons. So these are long, kind of tube-like things that the blood goes into the center of the tube, and the kidney cells reabsorb almost everything back again. And it seems like a complex way of doing it. Why would you make it so complex? Well, the main reason is if you excessively take something, then the transporters in the kidney are they're overdone. So it'll just simply go out into your urine. So it's a complex way of doing that, but for a free living animal, that's kind of important, because otherwise you'd have to be very careful what you eat and drink. Then what other? So we have your muscles. They are skeletal muscle. So they contract at will, unlike the heart, which the heart contracts based on the pacemaker. And then what's very important, people think, I think when I was a kid, I thought bones were just for structure, but inside the bones is basically where all your blood cells are made in the bone marrow. And then you've got the sensory cells, which are sensory organs, which are kind of complex, like the eye and the ear. And there you've got this little hammer thing that bangs off the drum. So it basically feeds in and amplifies sound. And then that's picked up by neurons. And then the brain there's loads of different cell types in the brain. It's not just neurons. It's Max, that's your specialty. Go ahead.
Yeah, you have different types of what is called glial cells, and these glial cells are the one which are surrounding the neurons. Actually, the neurons themselves are something like no more than 10% of the cells in the brain. And the glial cells are probably 70% of the other cells in the brain, and there are a few types of them. The astrocytes, which, as the name tells, looks like stars, which is why they are called astro. And the oligodendrocytes, they are the one which are surrounding the elongation of the neurons. And
what's the function of the astrocytes and the oligodendrocytes?
So basically, astrocytes clean the mess and provide sufficient nutrients for the neurons. So
they make glucose, don't they? I heard.
No, they don't make the glucose, but they provide a decent environment for the neurons to work.
I thought they were capable of gluconeogenesis.
They can do neoglucogenesis. Yeah, they can.
Yeah.
But that's not their kind of main function, in my opinion. They just keep the neurons in the best shape for working, which basically means that the neurons does what it has to do, but don't clean the mess. And in particular, the neuron works by transmitting information, and this is mainly done by neuromediation, secreting something to another neurons, achieving an action. And when they secrete those stuff, which are neuromediators, then you have to at one stage remove them. And this removal from it is not done by the neuron itself. The astrocytes are doing the job for that. And there
I
are
heard that
also
one astrocyte can touch thousands of other neurons, right?
thousands.
Yeah.
Uh,
Well, I read it on a wiki,
yeah,
so
it's I don't know.
but
Might be.
lots.
Yeah, definitely a lot. And it's every single neuron has its own kind of network of companions that works for him. So it is assumed that every single neurons has a capillary within 10 micrometer, which basically means that so the capillary is a tiny blood vessel, and then the complete unit which is around it is there to serve the neuron, brings it what it needs, make sure that everything is at rest for it, that there is no perturbation, so that the neuron can do its job properly, cleaning the mess, doing the dishes, making the environment clean for it. So
Is it, if Max, if you think a lot,
yeah,
will you use more energy?
yes, you would consume more glucose.
Are you sure?
No. No, but this is how this when you see this heat map of people and their area of the brain being activated, you see this. It's kind of popular. You see that on movies and things like that, where people have these helmets, and then you see their brain activities in specific locations, and that's how we know which area of the brain is responsible for something. This is mainly based on glucose consumption.
Yeah, but
So the
isn't it
assumption
true? 'Cause, I mean, you see all these silly films that say that you only use 2% of your brain.
Exactly.
Which is nonsense.
Yeah.
You use all of your brain all the time.
Limitless. This is limitless.
Yeah. So basically, you use all of your brain all of the time. But you can grow parts of your brain.
But what is true is that some psychedelic drugs kind of activate a lot of those cells. So probably doing abusing of psychedelic drugs means that you would consume a lot of glucose from your brain.
Yeah, but I actually saw that it reduces the synaptic interactions, drug abuse. So it's actually bad for your brain. So we just want to get drugs are bad.
That's your sensible. Your sensible suddenly pops up and say, "Oh, maybe I should say something on the podcast to promote doing drugs is something not to encourage."
Max, your mic is still a little hissing.
Oh yeah, sorry.
Well, I still want to go through a few more of the cell types. So you're pretty much an expert in brain parasites, I would say. But actually, before we go to parasites, I'd like to talk about microglial cells because I find them fascinating.
Yeah.
So microglial cells are cells that move around your brain, but it's even a bit more fascinating than that because the cells in your blood called macrophages, and they basically hunt.
So there are monocytes in the blood.
Yeah. And then the macrophages exist in different forms. So there's resident macrophages in the liver, for example. They're called Kupffer cells. And then there's resident macrophages in the lung. And then there's the circulating macrophages. And they basically eat up particulate matter. So things that just little things that come in, but they also eat bacteria in an unscheduled way. If they don't recognize it, they just eat it.
It's not fully true.
Go ahead then.
No, they do have some receptor for it. So these receptors are not extremely specific, but they do have a receptor for it.
Yeah, but I mean, it's not part of the adaptive immune system.
Yeah, that's part of the innate.
It's part of the innate is the ancient immune system that just recognizes foreign particles. It can't really learn, although that's probably not exactly true either, but just as a sort of textbook thing, it's
it's just that for a very long time we thought that the innate was not specific at all, whereas actually it is. It's just that they do have several receptors, and those receptors have a large specificity. Which makes them able to interact with a lot of bacteria, but they are not specific. Like antibodies would be specific to a specific virus, and we've heard a lot currently about the vaccination against COVID.
Yeah, but anyway, back to the microglial cells. So in human development, we think at least that some of these blood-borne macrophages get caught in the brain before the blood-brain barrier closes, and then they just differentiate into microglial cells, and they stay there for their entire life in the brain. They're not renewed. I guess they must be able to divide at some stage, but the pool you get is the only pool that we think, at least. And they go around and they clean up the brain of brain debris. If a neuron dies or something, they eat it up. And where do they put it, Max?
What?
Where do they put the debris? I guess they just chew it all up.
I don't
Recycle
exactly
it?
know. I guess they can, in the end, put it in the CSF, and then from the CSF it gets back to the blood. But I'm not really 100% sure of that.
Yeah, there's a lot of things.
And sometimes they can simply not get rid of the stuff, so then they form specific. Yeah, in specific neurodegenerative disease, you have a very specific hallmark of it, which you can see when you slice the brain and you stain it. Then you see this location where basically the microglia are activated. They are trying to digest everything, but they don't succeed in cleaning the mess. So then you still see the location of it. And that's quite common in some neurodegenerative disease to see these hallmarks of neurodegeneration.
And so we got all these different types of cells, and none of them last your whole lifetime. I think not except maybe neurons, do they? I don't know. I'm not sure about neurons.
Yes, they do.
Yeah, neurons must. The sensory neurons must.
Yeah. Because for a very long time we were thinking that neurons were not kind of regenerating. When I was young, I was told you have only a stock of neurons, and then if you do stupid stuff, then you're going to lose some, and it will never regrow because there is no regeneration of neurons. It's not fully true because we know now that there is neurogenesis even in elderly. So you are still capable of generating new neurons, but the fact is that these new neurons are not replacing the dead one. So if you lose,
not fast enough.
No, and it's also so much specific. This when your connection established during your childhood, progressively those interaction between your two neurons, and we know how that works. Initially, the cells don't have this kind of connection with one another, and they start. Basically, they give their hands to one another, and then the more you shake the hand, the more you reinforce this connection, and in the end, the other connections are going to disappear, and you're going to just keep the important connections, the ones that works when there is a feedback. And once this connection is established, you cannot replace that. So if you lose the original neurons, even if you have new neurons there, it will not take the place which has been left empty by the dying one. And that's where the issue.
This can be seen quite well in memory loss, right?
Yeah.
That if it's permanent memory loss, you simply can't go. I mean, you can form the original memory again by being told it, but it's a new memory of being retold an old thing. That's why memory is so fickle. I mean, I remember, I can't remember exactly what it was, but I've heard stories of people who swore their first bike was blue, and then they've shown a picture and it's actually red. Because you remember it and then you forget and then remember it again, and the more you tell stories, the more you make up memories. But what's fascinating about the whole thing is that the DNA. Well, there's caveats to all of this, but basically the DNA is the same for all the cell types. I think we can say it like that. So let's say it's exactly the same, but there is certain times where it isn't. That same DNA can make those 200 or so different cells, and this is something we don't fully understand. We know it quite well for the immune cells because they do it all the time, but in the resident cells, like say kidney and heart cells, they kind of just do it once in development, and that's such a tricky process that we don't know how that happens. And Max and I now are working with many other people in the field, but because we find it so interesting, we are working with these things called induced pluripotent stem cells. So you can take any cell in your body out that has a nucleus, and you trick it into becoming a stem cell, and then you can relearn it to be another cell by giving it different growth factors and doing different things. So Max is trying to make brain endothelial cells. I'm trying to make, together with my team, renal cells, and other people are doing neurons and heart cells, and it works quite well. I mean, it wasn't that this was fully original research, because people have been doing with embryonic stem cells for 50 years, but it's quite interesting that the cells in your body never really decide to change. They stay. They're happy being what they are, and we don't really know why. There's a cell type I was interested in back 20. Well, I'm still interested in, but I was doing active research in it about 20 years ago. It's a cell in the kidney called the thick ascending limb, and I was
What's
interested in it's the thick ascending limb. It has a specific function, but the interesting thing is it produces a protein called uromodulin that is secreted into the urine, and its function is nearly 90% of it is actually in the urine. And you produce 50 milligrams of this a day, which is really a lot. It seems like an awful lot of work to produce a protein just to be flushed out.
Yeah.
But it seems to be very helpful to keep your urothelial tract and your bladder clean from bacteria and to stop stone formation.
So this is supposedly the reason for it to exist, to basically prevent infection from.
Yeah, that's what we think. And every single cell in your body has the gene for that, and only one cell produces it. And there's no analogies to it either. So it's a unique protein. But I mean, there's many examples of that. The hepatocytes produce these plasma proteins, and that's unique to them. No other cell does that. I mean, they don't do it. So how does the cell know what cell type it is, and how does it know how to get there? I mean, these are still massive mysteries.
Who tells it what it should be?
Yeah, and then what they know, the cells also know when they're messed up, and they can die and cease to exist for the greater good.
Yeah.
So there's a thing called apoptosis, which is it's just a way the cell dies that can repackage its nutrients basically to the other cells without causing a big mess. And then, well, that's sort of the process of aging as well, because you've got these biological clocks at the end of the chromosomes called telomeres, and every time a cell divides, it shortens a bit, and once it's a certain length, a critical length, the cell doesn't divide anymore, and then it can still work, but it won't divide anymore. So that's basically the aging process, that they've divided as much times as they can safely divide, and then they stop dividing, and then you stop to repair your organs and your skin. And
is there any proper logic behind the etymology of the different cell types? Because I can think of some for endothelial and epithelial. So probably historically we had very few tools to look at the human body, except that we can easily identify the nature of blood, and the fact that it can flow was dating back from the antiques. And from that, you knew that there was blood inside the body and vessels, and then endothelial cells were the one surrounding the blood, so the one forming the vessels. And whereas as a position, the epithelial cells were the one in contact with the exterior of the body. So the skin, the inner cavity of the body. And then it's only by analogy that some others were discovered which were not really in full contact with the outer medium. But probably there is a reason for that. I know that you mentioned quite a few, but some other cells were named after their discoverer. but that was mainly because someone just
Yeah,
started.
I don't think there was any system. I mean, in the kidney, the proximal tuberous it just means they're closer to the glomerulus,
Yeah,
and the distal is just further away.
yeah, yeah, but they are
I-
specific cell types. I mean, they are part of a specific cell type. They are specialized epithelial cells, or they are specialized endothelial cells for the BBB, or they are specialized kind of type of cells. Whereas for others, they are specific to one organ, like hepatocytes are.
Yeah, but the hepatocytes are epithelial cells.
They are epithelial cells as well. Yeah,
But they're specialized epithelial cells, and they have an apical and a basolateral membrane as well. But just it's not just what we're used to in kidney.
I guess that's by analogy with the original epithelial cells, which were the one in contact with the outside, the inside of the body, which is kind of the gut lumen, for example, is considered to be something in contact with the exterior.
I think you have to remember that the first type of animals, they were like worm-like. So it was.
What's that?
They were worm-like. They were like tubes.
Yeah.
So it was simple: a tube inside, a mouth, and an anus, and then the inner. Well, over evolutionary complexity, like I guess humans, mammals are quite complex. Then all of these organs evolved in between.
Yeah. So, cells.
Cells.
So midi-chlorian. Have you ever heard that term, the midi-chlorian?
Midi-chlorian?
Yeah.
No.
So the midi-chlorian is supposedly the one which are responsible for the Force, which is in the Star Wars universe, the one that both the Jedi and the Sith Masters. And if you look at the terminology, midi-chlorian is actually a combination of the term mitochondria and chloroplast. And this whole theory is based on the endosymbiosis that you mentioned previously between an archaeobacteria and another cells, which embed something into it. So I guess George Lucas at that time had some biology class where he heard about this endosymbiotic phenomenon, which is making cells getting either chloroplasts or mitochondria. And inspired by that, he developed this concept of the force being mediated by midi-chlorians. The idea is that another analogy here is that mitochondria and chloroplasts are both tiny organisms in symbiosis with every single of your cells, responsible for generating some energy, and energy is force. So then if you look for a source of force within the cells, then you should have something more into it. And that's what the midi-chlorians are. So if you look back at episode 1, which is the
best
first episode. No, episode 1 is the first episode of the what was called the new trilogy at the time it was released, but the second trilogy, that's when Anakin is being tested for the number of midi-chlorian, and he happened to have a very huge amount of midi-chlorian inside his blood. So this is probably a bit of a mistake here, because probably if you expect to test blood, you probably mainly find red blood cells here, and they would not particularly be the best one to look for symbiotic within cells organoids. But anyway, it's science fiction, it cannot be perfect.
Yeah, it's far from perfect.
Yeah, but anyway, Anakin is being tested for it, and he has a number of midi-chlorian which is exceeding every single recorded ones, including Master Yoda's one, which is why supposedly it's that strong.
There is a lot of genetic mitochondrial diseases that so the mitochondria has its own DNA. It's differently structured than what's in the nucleus. It looks like a bacterial DNA. It's circular. All of the mitochondrial DNA is important. Non-functional mutations in that, which can arise in the oocyte or can be inherited as well from the mother, then there's lots of pretty terrible diseases that are at the moment very difficult to treat. I think they've even been talking about, I haven't followed this up really, but they're talking about doing mitochondrial replacement therapy. I think that would be done at the egg stage though. It's a pre-fertilization. Yeah.
Well, yeah. Then it's gonna be something deviating on something more ethical. But what would be the whole point of that?
That you can have kids, healthy children.
Yeah, but can't you simply substitute the egg by the egg of someone else instead of substituting the mitochondria for the sake of keeping the DNA?
Well, I should have checked this before I say it on the podcast, but I think wasn't this the case where there was children born with 3 donors? So the egg, which has the mitochondria, and then they took out the nucleus and put in the other nucleus, and then the sperm cells from the father donor, and then basically the child had 3 donors. I'm pretty sure that happened or happens.
Yeah, I guess everyone has his own takes of transmission through genes, but to me, you can probably adopt a kid and have the exact same benefit of being a parent. So I just this kind of idea of treating someone by substituting the mitochondria at the egg stage looks to me quite strange.
Yeah,
to develop so much effort.
I think it's probably even not that complicated, but it will be possible, and it's possible now to genetically modify an egg, I guess, quite easily.
It's a related question is whether we want to do so or not.
Yeah,
like ethically, because this is clearly the frontier with eugenism. When you can start touching the genes of an individual to be born, you can explain that you're going to do it for the sake of preventing a disease, but preventing a disease is very close from you're going to do it to make this new individual more powerful or having more strength or having the right eye color or whatever. And it starts being very touchy when it comes to ethics.
Well, luckily we have ethical boards and stuff like that, so you can't just play around with the genome. Yeah,
because I agree that it's probably just a question of money and effort here. Technologically speaking, it's probably not so much of a big deal to achieve modification inside an embryo by just touching the genes at the egg stage. But the question is, should we do it? That's different.
Yeah, that's what ethics are for.
Yeah.
So we can do everything, but what we don't need to do everything.
Yeah, exactly. It's not because we can do it that we should do it.
Exactly. Yeah. I'm not sure how much we've useful there, Max, to be honest.
Oh. So how much have we recorded now? I have no clue.
I think it's just an hour, but
yeah.
I wonder how good that is.
I have no clue. But maybe that's good enough. We can just stop with that and see what it looks like.
I'm not sure about the some of it.
Because I don't know why, but we suddenly started thinking that probably some of the people which might be listening to it might not all have the same background in terms of biology. So we needed to adjust some basic. Meanwhile, we cannot ban ourselves from going into a lot of details, which probably even with the set basics, they wouldn't really follow up. So it's
Let's try and rejuvenate it again. So it's not take two. See if you can knit some of this together.
good.
So the first thing, if you teach like I do, I teach a biochemistry course to well, it doesn't matter, but it's the chemistry students. And the first thing people try and address to non-biologists, really, because I don't think biologists ever think about it too much, is what is life? So how do you define it? I think it's a bit of a ridiculous endeavor. But if you look at a textbook says things like I'm just reading this out now. So they're highly organized compared to inanimate objects. They can do homeostasis, which means they can keep a relatively constant flux of the internals of the cell. They can reproduce themselves. They can grow and develop from simple beings. They can respond to stimuli. They can take energy and matter from the environment and transform it. And they can show adaptation to their environment. So something that can do all of these things is supposed to be alive. I can think of several examples where a single cell doesn't do any of these things, or not. Well, not all. The only thing I can see. Well, if you take
when you say single cell, do you have in mind single cell from a multicellular organism, or do you?
No, I don't. No, I mean that's the definition of what a living thing is.
Yeah.
So it could be a single cell or an organism, and maybe in fairness, with an organism, it doesn't have to be every cell in the body, because like the red blood cells are an example where they cannot reproduce themselves.
I think, as you once said when we were discussing a kind of similar topic, if you chop off your finger, the cells which are in the part of your body which is no longer attached to it is no longer a living thing.
Well, they will die eventually. They're living at the point of cutting it off, and for quite a while afterwards.
Yeah, what I mean by that is not every single one of your cells should be considered living organisms.
No, that's true. So a multicellular organism such as us, it's the whole being should be considered. But if you take it to the nth degree, and think about all life forms, all yeah, which so basic definition of life is something that everybody agrees on. It's a single cell or multicellular organism. So in a way, defining what life is defining what a cell is. But in my opinion, a cell, the unifying thing is that it can reproduce itself. It can multiply. You're laughing. Wait.
Yeah, because I just realized that I probably spent at least 8 years studying life science, and now probably 10 years teaching life science. And now I'm struggling really giving a definition of what is life.
No, because I think it's complex, and those sentences or those points I read out that don't really excite, if you put them like that. I mean, what is for sure is cells take extracellular substances and use that to create energy. I mean, that has to be. I mean, that's one of the grounding forces. And because everything, like life evolves from a single cell, a very simple cell, and then split and then rejoined again. The definition of life, at least on this planet, is the ability to pass on that genetic information, because we're based on evolution. That might stop at some stage if everything fully stabilizes, which I doubt. But we're talking millions of years, or even maybe billions of years from now. But at the moment, the ability to not that the organism has to reproduce, but the members of that particular species or whatever has to be able to has to have the ability to do it. Because we don't know of any life form that hasn't changed since the beginning of time.
In the meantime, if you stick to this definition you just gave, it would basically encompass evolution within the process of transferring, which basically means that you would consider every single living form on this planet to be one.
Yeah, but that's a higher point, and I do feel that. I mean, it's a belief that it is. I mean, it is one ecosystem. I mean, if you think of the.
If you think of it, evolution is one way of passing the information anyway.
Yeah, I mean,
and it's also fit with the idea of adapting to this to the environment, which is one other point that you listed as.
So, I mean, without doubt, all cells have apart from in a single organism like the red blood cells, which don't have DNA, but all organisms have DNA and the ability to pass that DNA onto its progeny, either the way it is or changed. So, some organisms don't do sexual reproduction, so they pass it on, but it can mutate down the line and then change it to something different. I mean, people wonder, about evolution. There is no purpose to it really, but you have to fit into the ecosystem, or the ecosystem changes, and then life has to adapt to that. I mean, humans are very disruptive beings in our ecosystem. Will we adapt to change it back, or at least to stop it changing so much? Or will life around us adapt to our disruption? I don't know. These are tough questions. I do definitely believe we really have to, rethink about our toxic behavior. But just back to that, if I define, I'm not. There's no consensus here. In fact, I cause upset when I say this. If you define life as the ability to be able to reproduce. And the potential to evolve, evolution is not necessarily needed, then unfortunately you'd have to say that viruses are a life form. They miss the ability to be able to take in energy or produce energy, but they use other cells to do that. I mean, they're wholly parasitic. But they're a very simple form of life. And as far as we know, they've been around since the very beginning of life. And they force evolutionary changes in other life forms. I mean, it's a big battle. The viruses that attack E. coli, what are they called again?
Bacteriophage.
Yeah, the phages, and the bacteria always is adapting. I mean, it was the Nobel Prize this year in chemistry was for the discovery of CRISPR-Cas9, which is a molecular biology tool, but it was an ingenious system of bacteria. They have actually their own immune system. A single cell system has an immune system to protect itself from bacteriophages. It's really, really exciting and interesting to show that even very simple cells are really complex. And like human beings, as I said, we have about I'm saying 200, but I don't know exactly the number of different cell types, but they all work together. And each one of them is fascinating. And they all work together and they all share the same DNA. So it's strange that even, as you said, Max, even as a biologist, it's difficult to define such a thing that's satisfactory. So I think we have to really put viruses out on their own because they are strange. They don't really fit any of the things except the ability to reproduce and change. They mutate all the time. We see this with COVID. That it's just getting. I heard people saying smarter. It's not. There's no intelligence there. It's probability basically. Because you don't hear of the stupid viruses that had a mutation that made them dumb and then didn't grow. So they're not smart per se. It's just probability that they have a certain amount of DNA, and if this changes slightly, they become more persistent.
I don't think I don't recall really questioning whether a virus was a living organism or not. The way we usually see it is questioning whether virus is a cell or not.
No, it's not. It's not a cell because it's.
Yeah,
It's,
yeah, but in the meantime, we easily agree on the fact that the virus is not a cell.
Yeah.
And in the meantime, we define a cell as the building blocks of the tiniest living organism.
Yeah, except viruses are outside of that.
Yeah. So basically saying a virus is not a cell, while in the meantime saying a cell is the definition of a living organism, basically implies that you would then consider a virus a non-living organism, whereas it is.
I think, yeah. So I think that's the thing. What are we trying to say? So it's not a living organism, maybe, but it is a life form.
Yeah.
Just a very strange one.
Yeah.
And like we think viruses, I guess humans think viruses are just out to be pathogenic to us, but they're out to get all organisms.
Yeah, I still have to read the books that you recommended a few times called The Self Gene. But can it be that a virus is a kind of evolution of a gene, or maybe a set of genes in that case, who find a trick to pass on?
But just to clarify, I'm not a virologist, and neither are you. But Richard Dawkins in his 1974 thesis, The Selfish Gene, he made a nice twist on life. He said that cells are just a vessel for DNA, and the not even the DNA itself, a single gene, is greedy and wants to make more of itself. And that's impossible to disprove because it's basically a fact, but it's conveniently simple thesis. I think that was the point of Richard Dawkins, to make it that simple so it would be challenging. But it's a fascinating book actually, and it's been revised a few times now as well. I haven't read the last revision, and then there's the people who like mitochondria as well, and you could put it the other way around, that life that holds mitochondria, so eukaryotes, were basically a way that mitochondria could be very successful. And so you could have the Richard Dawkins theory of mitochondria as well, although mitochondria have DNA in too. So then you've got these two symbiotic DNA sets that are happy to work together to make more of themselves. Well, I guess that's our hour.
Yeah.
I'm not sure if that was helpful for anybody.
Don't know.
We thought we should release another podcast. Maybe anymore we should try and make it a bit more structured.
Yeah, we'll see.
Yeah.
Let's edit it and we'll see.
Okay.
I'm going to stop the recording. So thanks for listening. And
Yeah.
see you.
Ces demeures qui parlent de microscope. Ces demeures potes qui parlent de microscope. Ces demeures potes qui parlent de microscope. Ça parle de bactéries parce que c'est des grands amis. En tout cas, pour résumer ce qu'il faut savoir dans cette lettre. La science, nous on fait de la science. Nous on travaille pour la science. Pour la science.